Now I'm going to finish today's video by talking about the three proteins that are associated with familial hypercholesterolemia. Familial hypercholesterolemia. Now the first is going to be an absent or defective LDL receptor, or an LDL receptor deficiency. Now we can see how this would lead to high levels of blood cholesterol, because our chylomicrons and our VLDLs wouldn't be able to find the cells that they need via their ApoC2 signals, meaning that our cholesterol and triglycerides are just going to be floating around in the bloodstream, not making it to the cells, which means that other cells are going to continue to produce these triglycerols, trying to get it to the cells, but ultimately it's just going to remain in our bloodstream. The second cause of familial hypercholesterolemia could be an absent or defective ApoB100. Now recall that ApoB100 is the structural cell behind VLDLs. Also recall that VLDLs formation is one of the three key pathways for eliminating cholesterol from the blood. If we do not have ApoB100, or we have a defective ApoB100, we cannot synthesize VLDLs, and thus our ability to remove cholesterol from the bloodstream will be severely inhibited. Lastly, we can have familial hypercholesterolemia by a defective PCSK9 enzyme. Now remember that PCSK9 was used to inhibit LDL receptors. Now, LDL receptors Lastly, we have the PCSK9 enzyme. In this case, it's unique that the mutation might be a gain of function. Although very rare, it has been known to happen. A gain of function PCSK9 would lead to the inability of chylomicrons and VLDLs to be recognized by LDL receptors, meaning that their triglycerides and cholesterol would remain in the bloodstream and not be taken up by the cells. Also recall, though, that PCSK9 inhibitors are on the market and have shown a decrease in LDL levels because an inhibition of the PCSK9 would actually increase the amount of LDL receptors on a cell membrane.